Okay, so let's begin. Starting us off today on this series of the Beckman graduate student seminars is Colton Parker. He is a fifth-year graduate student in Justin Rhodes' group. He did his bachelor's degree at Knox College, and today he's going to be talking to us about sex differentiation in sex-changing fish. So I'll let you take it away. Yeah, thanks, Colton.
Hey, okay. Glad to kick things off. Thanks everybody for being here and for your attention. So, yeah, I'm Colton Parker. I'm a graduate student working in the neuroscience program under Dr. Justin Rhodes, and I'm going to talk about my research looking at sex differences in the brain of these sex-changing fish and how we're trying to connect that back to a bigger understanding of sexual differentiation of the brain. So the brain region that we are interested in our research is the preoptic area. The preoptic area is a region of the hypothalamus. And if you have heard anything about the hypothalamus as a part of the brain, you've probably heard that it is a really important regulator of a lot of automatic functions of the body. Things like waking and sleeping, stress responses, feeding and hunger, and also reproductive hormone cycles. All of these things are controlled by cells in the preoptic area and other cells in the hypothalamus. In the human brain, it's located in the small region here, sort of at the base in the central part of the brain. Now, if we were to open up the hypothalamus and break it down into these different components, you'll see that it's made up of a ton of different subregions. And we don't need to worry about all of these. What we're focusing on in our work is the preoptic area, which is the region that controls the functions of the gonads, testes, and the ovaries, their release of hormones, their generation of gametes, the sperm cells and the egg cells. All of these essential elements of reproductive health are controlled by the preoptic area. The preoptic area also controls sexual behavior too, and so it has this multifaceted role in regulating this kind of holistic process of reproduction, along with other functions as well. Now, sex differences in the preoptic area, sex differences in its cellular makeup, in its genetic makeup, are necessary to produce sex differences in gonadal function and in sexual behavior. So what that means is that in order to have proper reproductive function of the ovaries versus the testes, which are of course doing very different jobs in the body system, you need to have a sexually differentiated preoptic area to regulate that process. What do we know about sex differences in the POA? So most of what we know, of course, at the cellular and genetic level, comes from research in animal models, and specifically in mice and in rats. If we were to look at the profile view of a mouse brain right here, this is the front side and the back side, and we were to make a cut right about here from top to bottom and open that brain out to look at the cross section, you would see the preoptic area as a group of cells toward the base of the brain, right about there. Of course, it's not purple. It's really just a cluster of cells that are tightly packed and then broken up into smaller subdivisions. We can break the POA up into a number of smaller subdivisions based on cell types and functions associated with them. And two of those subregions have really important sex differences that have been very well studied. One of those subregions is called the AVPV, and that's a region that is larger in females compared to males. And then another is called the SDN, and that's a brain region in the POA that is larger in males compared to females. Now, the AVPV, which has more cells in females, is necessary for ovulation and for the luteinizing hormone surge that triggers it, making this group of cells and its sex difference essential for proper female reproductive health and physiology. The SDN, on the other hand, which has more cells in males, is involved more so in sexual behavior regulation. Now, in males, which are generally better understood in a lot of biomedical research, tends to bias toward male mouse models, and that's something that is turning around slowly, but historically it's been the case. In males, testosterone is the first domino that falls to trigger the developmental cascade that leads to a larger SDN and a smaller AVPV. Ultimately, it comes back to testosterone as the trigger for those developmental differences. In females, on the other hand, there is not a comparable hormonal trigger to drive the sexual differentiation of the POA. You might think it's intuitive, oh, it must be estrogens that are kind of providing the complementary engine for this developmental process, but that's not really the case, actually. And so as a result, this process is really not very well understood, and that has implications for our understanding of reproductive health at the level of the brain. Now, in order to gain new insight and get more traction on this particular problem, we in our lab are trying to develop new models and use new technologies. Now, in neuroscience and biological research, one of the really great ways to get a new make new progress on a problem is to establish new model species that can complement already well-established models. And that's what we're doing in our research by establishing the common clownfish, Amphiprion ocellaris. This is a fish that's a really good model for this particular problem because they do not have a genetic sex determination. Instead, female sex development has a trigger that we can manipulate in the lab. And they have a POA in the brain that contains many of the same cell types that perform many of the same functions. So a quick primer on the fish model then, because these are really unusual model in neuroscience research. So clownfish are these Pacific coastal reef fish that live around anemone hosts, and they live in these small groups around their anemone with a linear social hierarchy. So there's an alpha fish at the very top of the hierarchy, and that's the biggest, toughest fish on the block. And it is always the female. The next fish in line in the hierarchy is the beta fish, and that's a breeding male. And then all the further subordinate fish are also males. And social status determines sex in these fish. It's not a genetic complement, it's the social status. What that means is that if the female fish grows old and dies or is lost to predation, that beta fish is going to ascend up the social hierarchy naturally because it is now the only big tough fish left on the block. And as a result of that, it's going to change sex from male to female. And that involves changes to the gonads, and that involves changes to the brain. So what is going on in the preoptic area during this process? What are the sex differences in the POA and how are they changing? The way that we are trying to study this question is using single nucleus RNA sequencing. This is a really, really powerful emerging genetic technology that allows us to take the preoptic area, break it up into individual cells, and then from each individual cell, out of tens of thousands of brain cells, collect a full data set of every single gene that is being turned on or being turned off in that cell, thousands of genes. And with this huge matrix of information, tens of thousands of cells, tens of thousands of different genes being expressed at different levels, we can look for relationships in that information and collapse it across a number of dimension-reduced spaces that capture as much of that information as possible to map out individual cells and look for clusters and look for relationships among them. So in these plots, each individual dot is an individual cell. The colors represent clusters of cells that are similar in their expression profile of genes, and the distance between cells on this plot represents their similarity or difference in overall gene expression profile. We've already identified in this data set a number of sexually differentiated clusters. 11 of the 49 clusters are differentiated based on cell number, and we have them color-coded here. So in red are the clusters that are greater number in females compared to males, blue, greater number in males, and you can even see the difference in the density, for example, in these red clusters here on the female side compared to the male side. We've begun classifying these cells as different neuronal subtypes, and we're going to further classify them based on peptides and receptors they're expressing. We can already see specific clusters of differentiated functions within these neuronal clusters that are sexually differentiated. We're also able to dig into this data set and evaluate brand new sex differences that we didn't predict initially at the outset. Things like this, where we identified one sexually differentiated cluster that is present in males and almost completely absent in females. And this cluster is highly expressing a number of reproduction-related genes, like androgen receptor, which responds to testosterone, and an enzyme that synthesizes dopamine, which we know is involved in regulating the reproductive axis, among other functions that dopamine has in the brain. And so we're going to further dive into this specific cluster to see whether it has a specific role in regulating differential function between the male and female phenotype, and then ultimately follow up to figure out how is it changing? How is it going from being very present in the male to being completely absent in the female? And what genes and what processes are driving that? And so our findings so far with the model are a number of potential female-biased POA cell populations that could stand in for the AVPV or something like it, and at least one really remarkably differentiated cluster that is expressing a number of reproduction-related genes that we didn't predict at the outset. And our next steps with this data set are to begin investigating more closely points of potential similarity and divergence between this data and available data from mouse POA to bridge the gap between this fish system and established models, mammalian models, and then also to study intermediate time points as we can track the change from the male brain to the female brain. And so with that, I thank you all for your attention. I want to thank everyone in the Rhodes Lab at the Beckman Institute, Beckman for supporting us, all of our research funding that has supported this work specifically, as well as our collaborators on this project at Georgia Tech who have really been essential in making this happen. And I'll take any questions you've got. Thank you.
Thanks, Colton. Do we have any questions for Colton? Feel free to unmute yourselves. If you can't, let me know. I'll go ahead then, I guess. Hey, Colton, so in the group of cells you showed on like whatever it was, the fourth slide or fifth slide, the difference between the male and the female.
Yeah.
When I guess it's the one with the plots. When the male goes to female, do those cells migrate or do they die, or like this is a map of the POA, right? Effectively.
Effectively, it's a map. It's a map of cell types in the POA. So it's not an anatomical map, but it's a functional map, I guess.
So in the sex change, do cells just die or get repurposed and become large and move to say that other red area where the females had a larger population of?
So we don't know, and there's a lot of possibilities here. We're right now conducting an experiment to follow up on the possibility that new cells are being generated in the POA, which could not account for this particular sex difference where cells are being lost from the male to female phenotype, but could account for some of these differences in the female bias direction. Now, with this specific transition here, the possibility of them physically changing their expression profile could be the case, although I think it's notable that these cells are really kind of island unto themselves. They're not very similar to a lot of other clusters, and so they would have to change a lot to move from here to someplace else in the gene expression landscape. I think that actual death of the cells is probably more likely because cell death and proliferation are kind of the two main engines in brain development and differentiation that ultimately leads to sex differences in cell number based on a lot of other research and other models.
Awesome. Thanks. Do we have any other questions for Colton? I got time for maybe one more. Okay. Oh, Professor Kirsch has a question for you in the chat.
Yeah, so the question is, how computationally expensive is this? This being analyzing data. And so the the initial pre-processing and genomic alignment is more expensive, and we have to run that on a server side. But the clustering and evaluation of differential gene expression is something that I can do locally on a desktop computer that's decently outfitted, or even on my laptop if I want to set aside a couple of hours to let it run. The actual analysis itself is pretty streamlined, and we actually are luckily able to leverage a lot of really great technologies that are developed with the express purpose of making this as inexpensive as possible.
Okay.
Yes, the clustering is based on principal components. The initial clustering is based on principal components, and then and then using kind of a k nearest neighbor approach to develop specific communities within those cells based on their principal components.
A patient with multiple myeloma, and so are there?
Sorry, what's that?
Yeah.
Is that?
I think that was
Might have been an
someone
accident.
Yeah. All right, Colton, thank you very much. Awesome work.
Yep.
And so now we're gonna move on to Sara. Oh my gosh. Moshage?
Yeah.
Oh my gosh, I practiced and then I failed. I'm so sorry. Sara works in Professor Mariana Kirsch's lab, studying the bone development in horses. She did her undergraduate here at Illinois, and is now a 5th year PhD student here at Illinois. So she stayed. She loves it here, and yeah. So please take it away, Sara.
Okay. Thank you for the introduction. And just a heads up, my PowerPoint has been acting up a bit. I think it's assigned to updates from Microsoft 2016. So I might have to restart some slides somewhere. So bear with me. So before I get started, I just want to make sure everyone is on the same page regarding some basic bone structure. So using this human femur as an example, there are 2 main types of bone tissue. So cortical bone is this dense bone found in the diaphysis of long bones and around the edges of the ends of the bones. And it's the primary load-bearing tissue. And it kind of looks like these cylinders of bone sheets wrapped around each other and then squished together. So it's very compact. And the other kind of bone is trabecular bone, sometimes called spongy bone because it looks very spongy and porous. And so this is usually at the ends of bones, and it attenuates loads and transfers them to the cortical bone. So keep this in mind. So fractures are the cause of over half of the fatalities in racehorses. So some fractures can be fixed, such as the example shown here. But when horses weigh so much that they can't really spend much time not using a leg. We can't tell them to lay down on the couch for a month while their fracture fixes itself. So if the chance of recovery is poor, the horse is put down. And my work focuses on the 3rd metacarpal and proximal phalanx bones, which are part of this MCP joint. And these bones account for over half of these fatal fractures. And we know from postmortem analysis that these are fatigue injuries, meaning micro damage accumulates in the bones before final failure occurs. And there are some researchers working on methods of early identification, but we're approaching it from what if we can strengthen the bone when the horses are young and prevent these fractures to begin with. So an easy way to strengthen the bone is through exercise. But when during one's life is exercise most important? So we know that during the period of growth from birth to maturity, the skeletal system is changing very rapidly and sort of primed for adaptation. So there are studies in animals that have shown that exercise during this period of growth has a greater effect on bone health than exercise in mature animals. So another question we have is, how long do the benefits of exercise last for? So using baseball players, professional pitchers as an example, and looking at their upper arm, their humerus bone, they provide an in-body control because most pitchers only pitch with one arm. So if we look at CT scans from the middle of their humerus, and this column right here is active throwers. So you can appreciate the larger footprint, the larger cross-sectional area of the throwing arm, and this thicker cortical shell here, and it looks more dense. And then if we look at what happens in the years since training as professional athletes, by the time we get to 50 years since training, the benefits of cortical thickness are gone. So the thickness here in the throwing arm looks about the same as the non-throwing arm, but the overall area of the bone is still larger, and therefore it's a stronger bone. So these benefits of exercise at an early age are sticking around for a long time. But what about exercise in horses? So there have been a few studies looking at exercise in horses, which is pretty unusual to exercise very young horses. So there was a comprehensive study in New Zealand in the early 2000s where they took groups of young horses, and the exercise group was exercised 5 days a week for 18 months. And then the exercise and control horses went on to be race horses, and the researchers followed them the whole time. So if we look at the results of the CT scans from the middle of the P1 in the exercise and control horses, on the x-axis you have age, and on the y-axis is bone area. The exercise horses have significantly greater bone area by 12 months. But you'll notice there's a really big gap between these data sets. There's an 8-month gap in these scans, so we don't know when the benefits of exercise arose. We might not need to exercise the horses for 18 months. So we know that exercise when these horses is young isn't detrimental. In fact, these horses that were exercised as young horses or foals went on to race and had some measure of protection against injury, but it wasn't significant. So we still have questions about what's the best method to strengthen the bones in these young horses. And there are a couple of major factors when considering this. So the most important one is, at what age should we do the exercise intervention? So horses are usually born and stay in the same place for about 18 months or so before they're sold and they begin training for their future job. So we're looking at this 18th month period. And we also don't know how long the exercise intervention needs to go on for it to be beneficial. So the previous study I mentioned, they exercised for 18 months and they saw benefits of exercise at 12 months, but maybe they didn't need to work out for that long. Perhaps 2 months or 4 months is sufficient. We also don't know how long they need to be exercising for. So most horse races in the US are between half a mile and a mile and a half. We also don't know what speed the foals should be moving at. They can't reach the speed that adult horses can, but is speed really important or is distance more important? We also need to know like direction of loading. So is it sufficient to run in straight lines? Do they need to be doing tight turns as in figure eights or loose wide turns? So these are all factors to consider when trying to find the ideal exercise intervention to strengthen the bones and reduce fracture. And if we were to test all of these on live foals, that would take a really long time. So the gestational period for a horse is 11 months, and they're expensive and large. So it would be difficult to test all these different combinations in live horses. But what if we could assess the exercise intervention computationally? So we're hoping to do that using finite element models. So these are models based on CT scans of the horses. So we get the geometry as well as regionally specific material properties from the CT scan. And then we can apply the loads representing a different exercise intervention to the model. And based on how the model responds, we can make inferences about how the bone would adapt to that specific intervention. So in order to go from a CT scan to a model, we need something called a density modulus relationship. So this relates a measure of density. In this case, we're talking about CT density to a Young's modulus. And this is obtained from mechanical testing of bone samples, typically in compression. And so Young's modulus is the slope of the linear part of the stress strain curve. So this is obtained from the mechanical test, and it's a measure of a material's resistance to deformation. And this is an important input to the finite element model because it tells us how the model or how the element will respond to a specific loading condition. And so these density modulus relationships are specific to which bone in the location of the body, and they also vary depending on species. So for horses, we currently have one density modulus relationship from the mid-90s, and it was done on the MC3 bone, this long bone here. However, the mean age of their samples was 6.7, which is well into skeletal maturity for horses. And as I mentioned, we're trying to capitalize upon the changes in the skeleton during rapid growth. So we're really focusing on what happens under a year. So this relationship might not be valid for us. And they also performed their CT scans on samples that had already been cut out of the bone. And we know that the density of a region of interest in an intact CT scan is different than the density of that same region of interest if it's already been removed from the sample. So another thing contributing to this is horses grow so quickly that their cortical bone actually is plexiform bone for a while. So it kind of has these like sheets versus her virgin bone. This is the cartoon I showed earlier. This is adult bone. You can kind of see the cylinders here. So eventually the cortical bone converts from plexiform to haversian, but we think that these might have different material properties. So we hypothesize that horses under a year of age will require a different density modulus relationship. And something that's really important to our development of a density modulus relationship is using clinical CT scans, because we want to apply this relationship to live horses. And to do that, we need to use a clinical CT scanner. We couldn't possibly fit a live horse into a micro CT scanner. So something that comes along with that is reduced resolution of our image and partial volume effect. So this box represents the outline of one voxel, which is a 3D pixel. And this orange area is the actual bony tissue. So if this is what the bony tissue looks like in real life, if we were to look at it on a CT scan, it would end up being averaged out. And so through this partial volume effect, multiple different configurations and densities of bone will end up looking the same. And what that looks like on a real sample is a micro CT on the left and a clinical CT on the right. You can see we lose a lot of information. You know, there's still important stuff there, but the benefit is our horses stay alive, and we can scan the same horse at multiple time points while it's growing, which is really beneficial. So how do we get this density modulus relationship? So it starts with a scan of the whole bone done on the same scanner that we use for the live horses. And then I take micro CT scans of the bone and merge them into one data set. Then I cut the bone sample into slabs and use a drill press to remove cylinders of bone. And these samples are then tested in a compression machine. And the main output from this is the Young's modulus of that sample. Then I take these slabs with the cores removed and scan them again, and then locate precisely in the intact scan where those cores are. And then I transfer that location over to the clinical CT scan. So in this case, I'm only using the repeated micro CT scans to locate where my cores are. And then we can get exact information about the clinical CT density of the cores from an intact CT scan. So this is what it looks like. On the x-axis is ash density. So this is a measure of the mineral in each sample. And each dot here represents one cylinder of bone that was tested. And the y-axis is Young's modulus, the main output of our mechanical test. So when we fit a curve to this is our density modulus relationship. And when we look at the one from literature, which is in gray, this is what I talked about, the density modulus relationship from adult bone, we can see that it over predicts the modulus. So right now, from this set of data, we can say that we need a different density modulus relationship for growing bone. But we still have more work to do. So right now, this is a combination of samples from the MC3 and P1 bones. So eventually when we test more samples, we will be able to see if those two bones require different density modulus relationships to model them. And we'll also look at cortical versus trabecular bone. Right now, this is all trabecular bone, as well as some other factors. But the end goal is to have a density modulus relationship or multiple ones that apply to horses younger than a year. Sorry, I'd like to thank my lab group and my advisors, Mariana Kirsch and Annette McCoy. And thank you to Beckman Institute for their 2020 research fellowship, as well as all the resources there. So all my micro CT scans were done on the Ragaku CT lab scanner. So thank you, Lele, for all your help, as well as the Viz lab, which is where I do all of my computational work. So thank you for your attention.
Thank you, Sarah. That was awesome. Do we have any questions for Sarah? Feel free to chime in or raise your hand and mute yourself. Okay, Sarah, hey, based on Colton's last talk that he just gave, do you see a lot of differences in in behavior of bone density between males and females?
So far we haven't, but we also don't have enough information yet to confirm that. The samples that I'm looking at so far, they're all under 6 months old, so they're not their sex hormones are not really activated yet, so we don't think there are differences. That is something that we can look at once I have more samples tested. We can look at the effects of sex, but right now we don't have enough information to say so. But my thought is that at this young of an age, no.
Okay.
So Mark Taylor asked, how exactly do you exercise a horse? I don't have a video here, but actually this guy right here, his name is Cash. We ride him and lead the moms. So they're in the background here, and then the babies follow the mom. So that's how we exercise them here.
Great. Well, awesome work, Sarah. Do we have any more questions? And if not, I don't see any. We're going to go on to our last speaker, Edward. So Edward, go ahead and share your screen. Edward is a fifth-year PhD student in Charles Schrodis's group. He did his undergraduate at Cleveland State University, and he's going to talk to us about some chemistry, doing some automated chemical synthesis and conductance measurements on molecules. So take it away, Edward. I'm excited to hear what you got to say.
Thank you. Hello, everyone. So today I'm going to talk about some of the work I've done developing automated synthesis for kind of an accelerated research and discovery for organic electronic materials. So organic electronic devices are essentially just electronic devices based on electronically active organic molecules. So these can consist of things like organic solar cells, organic redox flow batteries for grid scale energy storage, and organic transistors for flexible electronics. And you can see some of the example chemical structures there on the bottom. They're generally these kind of organic structures with kind of these alternating single and double bonds that are what kind of grant the conductive properties to these materials. And in general, these devices, when you compare them to traditional inorganic electronics that rely on things like silicon and vanadium, these organic electronics promise to be, a lot lower cost, easier to process, more tunable because you can kind of tune that molecular structure to tune the electronic properties, and easier to, they're printable from solution. So they pose a lot of advantages, but in terms of the actual electronic performance of these devices, they still generally lag behind their inorganic counterparts. And a large reason for that is we kind of lack kind of defined rules for how we can tune this chemical structure to tune the performance of these devices. So as you can see from some of these example structures, you can imagine the chemical space and the number of different structures you have to work with is really huge. So it's really, and the studies that have been done to this point are really just based on a trial and error approach of predicting, based on intuition, what structures might work well, and then trying to synthesize those and test them. So we really want to develop more concrete rules for how that structure of those organic molecules impacts the function of these electronic devices. So we're thinking about things like the length of these molecules, swapping out different heteroatoms, changing the positions, looking at adding different side groups and how those affect things like the conductance, the packing of these molecules in a basically solid film, and the optical properties, how they absorb and transmit light. And in this talk specifically, I'm going to highlight an example looking at how the length of side chains appended to these molecules will affect their conductance at the molecular level. So to understand why we're interested in side chains, these solubilizing side chains, as you can see on these molecules on the bottom left, are kind of ubiquitous and very necessary for these organic electronic molecules. Basically, having these side chains imparts solubility in these molecules so you can dissolve them in some solution and then do solution printing onto some device, which is how these devices are generally manufactured. However, often very limited consideration is given to how these side chains might actually affect the device performance beyond granting that solubility. And there are a few examples to show that these actually do have a dramatic effect on the device performance. So this is one example where they just had this oligothiophenol molecule, a very common organic semiconductor, and they varied the length of the solubilizing side chain from 4 to 8 units. And they see that the performance of this molecule in an organic solar cell actually drastically changes based on the length of the side chain. And they find that there's actually an optimal side chain length, which is 6 carbons. So this really highlights the importance of these, kind of understanding how these side chains affect electronic properties for these molecules. However, there's kind of 2 key challenges that we want to address moving forward from this. And one is that these are device scale measurements. So the side chains, it's kind of hard to decouple the effect that these side chains have on how the molecules kind of arrange, next to each other in the film, and then how a single molecule, how the side chain affects the properties of a single molecule and how it affects how that single molecule interact with other molecules. So it's very complicated, it's hard to get at the fundamental effect the side chain is having when you have this bulk device level property and you're looking at things from a very high level. And then also, these types of studies are generally really hindered by synthetic challenges. So basically, generally for every new molecule you want to test, every study you want to perform, you're doing a specialized synthesis that's specific to that molecule. It can be very time consuming and specialist dependent. So we want to look at, can we develop more general, easier synthesis methods to access a wider array of these organic molecules. So to address the first point and kind of probe the effect of these side chains in a more fundamental level, my group specializes in single molecule conductance measurements. So this specifically is a technique called scanning tunneling microscope break junction, or STMBJ. What this device basically consists of is a gold substrate and a very fine gold tip. The tip can be moved up and down to repeatedly break and reform contact with the gold substrate, and you can apply a bias between these and measure the conductance through the tip and substrate. So what you see on the bottom left is we're measuring the conductance as a function of displacement as we're moving this tip up and down. At low displacement, the tip and substrate are in contact, so you have very high conductance. However, as you move that tip up, you eventually break the contact between the tip and substrate, and the conductance value drops off below the noise level of our instrument. When we want to actually measure the conductance of a molecule, we place a solution of our molecule that we're interested in testing on the substrate. And we can look in the second plot on the bottom, as the tip slowly moves up, we get that breakage of contact between the tip and substrate, which drastically decreases the conductance. But instead of dropping straight to the noise level, we see this plateau here. And this plateau actually represents a single molecule being trapped between the tip and substrate. So we're actually directly now measuring the electrical conductance of a single molecule. So we can look much more fundamentally at the electronic behavior that these molecules have. And for each molecule that we test, we perform these measurements thousands of times to gain a statistical distribution. We can see the 1D conductance histogram that's just looking at the conductance value over many measurements. And then we also can construct these 2D conductance histograms that also show the displacement at which we're measuring this molecular conductance, which gives us an idea of the length of the molecule in that junction, which gives us some clues into its conformation. So to address the second point, which is how can we access large libraries of these materials for our single molecule conductance measurements, we leverage building block-based iterative Suzuki coupling, which is kind of analogous to iterative peptide synthesis or nucleotide synthesis, which are much more established technologies. And we basically encode whichever functionality we're interested in into these bifunctional building blocks. In this case, we use haloboronic acids. Suzuki coupling is a coupling between a boronic acid and a halide. So by having these bifunctional haloboronic acids, we can couple them together sequentially. And essentially, this specific technique leverages this meta-protecting group that covers up the boronic acid and prevents it from participating in the reaction until we're ready. We can remove this protecting group under mild basic conditions to couple it with the halide of a subsequent building block, and then basically repeat this cycle over and over again to build up these precisely sequence-defined molecules one by one. And you can see just one example of what these molecules might look like on the bottom right, where basically each color represents a different building block that was stitched together to make this larger molecule. So with this technique, because we're kind of performing the same operations over and over again, and it's a very robust technique that works with a lot of different building blocks in the same conditions, this kind of really naturally lends itself to automation. So we developed a automated small molecule synthesizer. So this is a kind of a second generation instrument. It's fully automated, controlled by custom software that performs just iterative cycles of this deprotection to expose the reactive boronic acid, the coupling reaction between the boronic acid and the halide, and then a purification between each step to ensure that we have high quality products moving through the system. We've done a lot of work to increase the parallelization, so we can do up to 16 reactions at a time, and we can adjust the scale, and it takes typically 12 hours per reaction. And so this has become a really powerful tool that we've been able to leverage for a lot of different studies, including the sidechain dependent study that I'm going to talk about in a bit more detail moving forward. So now that we have kind of our measurement technique, as well as our synthesis technique, we want to actually think about how we're going to design the molecules that we're going to use to probe the effective sidechain length on the chemistry or on the conductance of these molecules. So we need to choose some kind of conductive bridge. So we need some kind of bridge for the molecule that will grant it enough conductance to give a robust signal in the measurement. And in this case, we choose just phenyl, which is a very simple and reliable, robust conductive bridge for these type of measurements. And then we also need some kind of anchor, which will give our molecule basically a stronger interaction, either dative or covalent interaction with the actual gold electrodes that will make the junction of the molecule sitting between those two electrodes stable enough to perform, to give us enough time to actually perform our measurement. And in this case, we're going to choose these thiomethyl anchors, which have lone pairs that can interact with the gold electrodes and help us form a very stable junction. And so we can encode both the thiomethyl anchors, the phenyl backbones, as well as our side chains directly into building blocks that can be plugged into our automated synthesis method. And using the parallelization of our method, we can develop a library of these different terphenyl molecules that have the anchors necessary for single molecule conductance, and then pre-installed side chains of varying lengths that can allow us to look at the effect of solubilizing side chain on the conductance. So we developed a library of these terphenyl molecules with side chain length varying from 0 to 12. So these R0 represents side chain with 0 carbons, so no side chain, and then R12 represents a side chain with 12 carbons, and these are all linear side chains. And we measure the conductance of all of these at the single molecule level. And what we see is very surprising. So for R0 up to R4, we see just one conductance peak, which is exactly what we'd expect for these type of molecules, and it's conducting at a level which is similar to what we expect for these molecules. However, when we increase the length of the side chain, just one carbon going from 4 to 5, we suddenly see this dramatic jump in the conductance where we have this second peak emerge that's an order of magnitude higher in conductance for these molecules. And this is very unexpected that side chains would have this dramatic effect, and specifically such a dramatic increase in the charge transport both on a quantitative and qualitative level. So we wanted to do a few additional experiments to probe what was causing this. So we first hypothesized that we may have a kind of through space interaction in the junction. So normally a junction looks like what you see on top, where you just have one molecule that's spanning from the tip to the substrate. But you can also have a case where you have one molecule on the tip and a different molecule on the substrate, and then you're actually getting charge transport through space between those two molecules. And we thought this is something that might be facilitated by the longer side chains. So in order to test this, we can just use our building blocks-based synthesis and just quickly synthesize this derivative control molecule that has one anchor removed. And if we are seeing through space charge transport, we should still see should still see the same signals with this molecule that only has one anchor, because this through space charge transport only requires one anchor because the molecule is only bound to either the tip or the substrate. But instead, what we see is that this single anchor molecule shows a very low signal, nothing compared to what we're seeing for the two anchor molecules. So we can conclude from this that what we're measuring is actually a single molecule interaction in the junction. So another thing that we know about the charge transport in these molecules is that so we have two phenyl rings next to each other, and they're kind of free to rotate around this bond between the phenyl rings, you can imagine. And the conductance actually strongly depends on the angle of rotation between these two rings. As they become more coplanar, they have a higher degree of overlap between their electron orbitals, and that leads to higher conductance. And as they become more twisted, that conductance decreases. So we synthesized a variety of derivatives to kind of test if this was the origin of our high conductance peak that we're seeing. So we synthesized a molecule that's forced into a planar conformation due to these extra bonds here. And then we also synthesized a molecule that's forced into a more twisted conformation due to the steric interactions of these side groups. And what we see, we test these both experimentally and computationally, and we see that the conductance dramatically decreases as the twist angle increases. But importantly, what we also see is that the conductance of the planar molecule matches exactly with our high conductance peak for the side chain molecules. So putting this all together with some additional control molecules that I don't have time to go into here, we basically can create this picture of what's happening with the molecule that, normally when we see our low conductance peak, it's just the molecule is standing up on the substrate, tip comes down, and we have this molecule in the extended position. It's free to rotate, and we see a low conductance signal because we have a high twist angle. But these side chains facilitate these molecules to absorb onto the surface, which enforces planarization in these terphenyl molecules, which leads the molecule to be trapped in this planarized state, which when the tip comes down, we're then measuring a fully planarized molecule that has an order of magnitude higher conductance, which is facilitated in these side chains. Just to conclude, this is, illustrating an example of how side chains can drastically affect the conductance properties of these molecules by affecting the interactions they have with the gold substrate and changing the conformation of the molecular backbones. In the future, we hope to extend this to a variety of different conjugated bridge chemistries to test the generality of this technique. I would like to thank a PhD advisor and my entire group, and specifically Nick and Professor Burke, who were really helpful with collaborating with the iterative synthesis, and then Song Song, who collaborated with us with the conductance measurements. So thank you all for listening. I'd be happy to answer any questions.
Thanks, Edward. Do we have any questions for Edward? Feel free to chime in or type them in the chat. All right, Edward, I'll go ahead. How do you, so in this entire presentation, you show basically trimers, right? It was one molecule and then connected to bridging two molecules that were connected to the gold surfaces, right? It was how can we think about like extending that? Can you like can you extend it beyond a trimer? And does that mean the conductance just drops off dramatically, or have you looked at that?
Yeah. So as far as extending it, because we have this iterative process, we can effectively do as many iterative steps as we want, and each step would add another block. So we could extend it. Our key limiting factor is, well, one, the solubility of the molecule as it gets longer. Two, the solubility, as you mentioned, will drop, but it won't, we could go to 4 or 5 probably and still measure it fine. And especially with this high conductance that we're seeing from the side chains, something we're really interested in is extending to longer molecules and seeing if we can observe conductance for longer than normal molecules because of this side chain interaction that we're having. So yes, we can extend it. We haven't in this study, but we would like to in the future.
Cool. Professor Kirsch also has a question for you in the chat.
So the question is, can you have looked into the effects of molecular geometry on the effects of the molecules, e.g. how stiff they are? We haven't specifically looked at those effects in this study, although there are studies with more flexible molecules that basically trap a molecule in the junction and then move the tip up and down to change the conformation of the molecule. And you see the conductance change for these molecules. But these molecules, we at least know from chemical intuition as well as simulations that they're pretty stiff as far as their only rotation is just the rings changing their angle relative to each other. They don't have much flexibility along their backbone. But that is something that's very interesting, within this field, is using the single molecule conductance measurements to look at the effect of probing stiffness and changing conformation.
Could you use acetylene linkages or just normal double bonds to look at that? Like, how does conductance change with those structural features?
So you're talking about like changing the actual anchor molecules?
No, the linker. So it's like if you, instead of having an aryl-aryl bond, you had an aryl-acetyl aryl, or an aryl, like a double bond, right? Because do you understand what I'm saying?
Yes. Yeah. So just having more flexible bonds in between phenyl rings. You're asking? Yeah, I think that would
still potentially conduct. Yeah.
Yeah, that would definitely be something to look at. When you do that, you're losing conjugation in most cases. So you would probably have lower conductance, but I mean, people have done this for even just alkyl chains. So there is possibility to, introduce some flexible groups in there and then see how that would affect these phenomena for sure.
Awesome. Like there's one more question on, I guess. Oh yeah, okay. Got it. Professor Kirsch just followed up.
Oh, I see. Yes.
Yeah, it's just a comment.
Oh, okay.
If you want to make it comment yourself or not.
Yeah, Professor Kirsch just added, it would be interesting to see how longer chain flexible molecules behave. And yeah, I totally agree. Especially with this, side chain phenomena, can they still achieve this planarization if the molecule has more flexibility, and how can you control that by kind of moving the tip up and down to push and pull the molecule in the junction? I do think that's really interesting to pursue. So thank you.
All right. Well, if there aren't any other questions, join me in thanking all of our speakers today. Great work, all of you. Thank you, everyone who came out. And yeah, we hope to see you at the next edition of these seminars. A lot of great, interesting work being done here at Beckman. Thank you, guys.